Vaida Salaburda. I am from across the square from Life Science Center, Department of Neurobiology, from the Department of Neurobiology and Biophysics, and it's a great honor to introduce this lecture, Zoltán Molnár. By introducing, I would like to say three things. First of all, it's Zoltán Molnár, amazing scientist. He made a very good research, publishes in most amazing papers, and he will talk about layer B6. I'm almost sure that you don't know what it means, so he'll tell about this. It turned out that this layer is important for regulating neuronal function, and as it regulates this functional and activity, it may be important for kind of diseases. So this is the first thing. I think it will be very important and actual topic. Next thing, just pay attention what is neuroscience nowadays. What kind of skills and technique you should use to make this things happens. What kind of techniques used and what you should do in order to assess all these brain functions. And the third thing I would address to junior listeners in the audience. Maybe you are sitting here and thinking, is it possible that someday I'll collaborate with such a scientist like Zoltán Molnár? And answer is yes, because I'm very proud. Our alumni, Augusta Vadishuter, she graduated neurobiology a couple of years ago in our department. She's continuing with Zoltán in his lab. So, nice, big honor to introduce you, and Zoltán, floor is yours.
Thank you very much for this very kind introduction. And when I gave this title, I didn't know that there will be this discussion about light and the shadows of God. So I think my title is perfect for the talk. This part is, the cis function is a spelling mistake. It's not, it's a dysfunction. So I don't know whether you have been to Oxford, but I will tell you that the word neurology, neuroscience, is coming from Oxford. So you already learned something. In 1664, Thomas Willis used this word neurology first. So we can consider Oxford as the seat of neuroscience. So as you know, the adult mammalian brain is the seat of higher cognitive functions. Our brain is about 1,350 grams on average. We have millions of neurons interconnected with trillions of connectivity, and all this is determined by about 30,000, less than 30,000 genes. So we have to have very complex interactions with the environment to form this complex structure where different functions are located in different parts of our brain. So for instance, when you are looking at an object, then this part of your brain is active in the occipital pole, where you have your primary visual cortex. And when you wiggle your fingers, then this part of the brain is active in the primary motor cortex. And if you look at the structure of the outside of your brain, the cerebral cortex, you have 6 layers of neurons in this structure. And these were actually divided up to 6 layers historically. So neuroanatomists decided, okay, let's have 6 layers, and then they kept using this scheme across the animal kingdom. But they could have had 7, 8, or 9. They could have justified that as well. But we ended up with 6 layers. And in these 6 layers, you have different cell types with different morphology, and with different connectivity, different size, different packing density, and the slight variations in the structure of the cerebral cortex is reflecting the different computational functions these cortical areas have to perform. So that's why in your primary visual cortex, you have more layer 4, which is receiving the input from the thalamus, whereas in the primary motor cortex, you have more layer 5 neurons, and these are the neurons which leave your skull, go to your brainstem and spinal cord, and then you can move your fingers. So the aim of my talk is to describe how these differences emerge and how do we put together a functional nervous system and what can go wrong. And by the end of the talk, I think you will really appreciate that you have a normal nervous system. And because so many things can go wrong, and that would hinder you in your academic progress. So the topics I will cover, instead of just the transient cells, which I originally wanted, but during listening to the talks today, I decided to broaden the scope of the lecture, is to talk about how neurons are born, how these neurons, which are born away from their final location, find their way to the target areas. How will your brain receive its folds? How will it accomplish this shape? What can go wrong? Also, what can go wrong with the interaction between the mother, the diet, or other agents which the mother is consuming, how it will affect the brain development, what can go wrong. I will talk about the fetal alcohol syndrome, some of the inflammations which can occur, Zika microcephaly, and these kind of conditions. I will show you that they can have a huge effect on brain development. And then, finally, I will talk about some of these circuits which are essential for processing information of the world. So we just had a discussion about light, but nobody mentioned that you need a functional brain to perceive light. No, there's no point having a retina. The brain has to listen and interpret this light. And you will see that all what we perceive is highly regulated by these circuits of the brain. And lots of things can go wrong, what are you interested in, what you find stimulating. Right now, half of your brain, and also my brain, are asleep. But which half is regulated by these loops between the cerebral cortex and the thalamus? So I will talk about what can go wrong with the development of these pathways. And finally, I talk about the transient cell group, the subplate cells, which normally have a very important role in forming these circuits, but then they largely disappear. And you only get just a few % of these cells remaining. Around 20% of them will survive to adulthood. But these cells have very important roles in regulating what information is reaching your cerebral cortex. And no wonder that in autism, schizophrenia, attention deficit hyperactivity disorder, you have an altered number and distribution of these cells. So I will link these transient cells to these cognitive dysfunctions. So I think we are living in a really exciting time right now. I would compare these decades today to the centuries we had in 1544, when people started to discover where the continents are. You had these very famous contributions to chemistry. Then also the heliocentric view of the world appeared. And this is the time when people started to look at the brain also. And before they were looking at the brain, this was the very common view of our brain. So they put in 3 ventricles into the skull, into the brain, and they were talking about this cell theory, that you had 3 cells filled with cerebrospinal fluid, and they said that these chambers were processing the information in the brain. And this drawing has been repeated by many, many scholars of the time without any criticism. Obviously, they never looked at the brain. They just looked at each other's publications, and they kept repeating it. Even Leonardo da Vinci, early in his career, he was drawing these 3 ventricles in his drawings. Obviously, he didn't look at the brain himself at that stage of his career. But then, a few years later, he grabbed an ox brain and poured some melted wax into it, removed the brain, and discovered that this is the real ventricular system of the ox brain. So you have the 2 lateral ventricles, the 3rd ventricle, aqueduct, 4th ventricle here. And you get a very similar image if you do it in a human brain, which we use to terrorize second-year medical students in the dissection room. So you can see that this is a resin cast of a human brain with the two lateral ventricles, third ventricle, aqueduct, fourth. So after Leonardo da Vinci was dissecting, he modified his view, and he also put the third ventricle into the middle of the cranial nerves that they were receiving this information. So anatomy was changing, and the change came from moving from this position to this. So anatomy was taught by a professor who was reading Galen's or Aristotle's work. He didn't look at the body. There was a special dissector, a barber, who was cutting the body. And then there was also somebody who was pointing at some body parts based on the literature the teacher was reading out. Obviously, it was outdated text from Aristotle, so everybody was confused, and the medical students didn't pay any attention to anything. That didn't change, by the way, even today, but in the dissection room. So what changed was, between these two drawings, that Vesalius, who was a leading anatomical figure, he actually moved next to the body, and he was dissecting himself. And he didn't believe anything what Aristotle or Galenus was writing. He had to see things with his own eyes. And that caused a revolution, because his anatomical description was completely accurate. And if you look at some of his publications, we could use the drawings even today. And then this is the time when you have all these anatomical theaters built in Padua, Bologna, Leiden, London. And then we started to get a much better understanding of anatomy at the time. So this is the time when in England, Henry VIII regulated medical profession. So there was a Royal College of Surgeons, and then these could actually claim the body of executed prisoners for dissection. So this is the time when Oxford had lots of colleges established. My own college, St. John's College, was established in 1555, and they had about 400 students already. So this is the time when my hero, Thomas Willis, was born, and now you know that Thomas Willis gave the term neurology, neurologia, to the world. And also, if you talk to medical students, they will all know the name of Willis, because the arteriosus circle is called the circle of Willis because of him. So he was dissecting with other scholars, including chemist Robert Boyle, anatomist William Petty. He was teaching Richard Lauer, who was his student, and Christopher Wren. When you visit the St. Paul's Cathedral in London, he was the architect behind St. Paul's Cathedral, and also All Souls College and many other buildings, Sheldonian Theatre in Oxford. So you see, interdisciplinary interactions are very important. So it's not just the open reading. They already did that in 1660s, these interdisciplinary interactions. And if you look at these names, you notice that all these were founders of the Royal Society of UK. So they were dissecting together, and the drawings of Thomas Willis were produced by Christopher Wren. So this is where he worked. So next time you go to Oxford, go to this street, Merton Street, and this is the Merton College Chapel. This is Corpus Christi College, and opposite you have this building which is called Beam Hall or Beeham Hall, and this is where Thomas Willis worked and lived and gave the term neurology. And these are the drawings I'm talking about, and this is the arteriosus circle, which is produced from the two vertebral arteries which come in through the foramen magnum. They unite into the basilar artery, and then they form this arteriosus circle with the two internal carotid arteries. And this is called the circle of Willis. So every medical student will know this. But very few would know that Thomas Willis was the first who suggested that the mammalian cerebral cortex is the seat of higher cognitive functions. He suggested this based on comparative data. When he dissected a sheep brain, he noticed that the cerebral cortex was smaller, less convoluted, but the brainstem was very similar. Even the circle of Willis was very similar, but in proportion, the cerebral cortex was much smaller. So Willis said that it's the cerebrum is the primary seat of the rational soul in man and the sensitive soul in animals, and it is the source of movements and ideas. And indeed, if you want to do language, episodic memory, complex physics, mathematics, you need cerebral cortex. Without that, you can't do it. His other line of argument was based on dissecting his own patients when they died. So he was a practicing clinician, and he was observing his patients during life, could see the symptoms, like epilepsy, learning difficulties, and when they died, he looked at the shape of and size of the brain, and this is a normal individual. Obviously, the brain is in a lamentable condition because they didn't use fixative. Later on, they used port wine and vinegar because port has very high alcohol content. But initially, they didn't. And this brain is from a boy who had signs of intractable epilepsy and learning difficulties. And Willis discovered that this boy had more but smaller sulci and gyri. And because of that, he suggested that the symptoms probably occurred because the boy had an altered cerebral cortex. And Willis got it absolutely right. Probably he was looking at a polymicrogyria syndrome, which is a condition where you have altered neurogenesis and neuronal migration of the cells, and they usually accompanied with intractable epilepsy and learning difficulties. So Willis was way ahead of his time in 1664 when he published this. And now we know that brain developmental disorders can cause all sorts of problems which manifest in cognitive dysfunctions. But today, I'm not going to talk about these very severe developmental abnormalities. I will talk about more subtle developmental abnormalities. So this is where we have to start. The entire nervous system is developing from a neural plate. And this neural plate is closing into a neural tube. And this neural tube will then form the telencephalic vesicles, the eye, the diencephalon, and also the spinal cord. And you will see that this rostral end of this tube will develop into the cerebral cortex. And it is really interesting how this structure will have lots of cells. They interconnect with the sensory periphery in utero. And then when we are born, you can start interacting with the environment straight away. So then you will get sensory-driven stimulus in a system which was built in an autonomous fashion. So at the time when we are born, within a few hours, you recognize your mom's face. In fact, even in utero, you recognize the mom's voice. So there's a lot of work going into the development of the brain before we are born. Most of our neurons are generated at these very early stages. And also, most of the connectivity is developing before we are born, but then myelination will go on until puberty or even later. And then some of the cells will die off, and you won't have any of these transient cells within the postnatal brain. And your brain was the biggest when you were about 11, 12 years of age. Now your brain is becoming much thinner. Mine is already very, very thin. But this was the peak when you had the largest cerebral cortex. So note that we can now survive even if you are born at 24 weeks of age. But note the huge work which is going on during this period until 40th week. So it makes a big difference whether you can finish this development in utero or you have a prematurity. So these steps now can be measured with biological markers. You can correlate the firing of the neurons with some landmarks of maturity, and it's a really exciting time to relate this to brain development. We humans have a big problem. We are born extremely immature. So when we are born, we really need care and attention for several years. So for those of you who have kids, you know exactly what I'm talking about. That if you have a turtle, this hedge turtle can run to the sea on its own with the nervous system which developed in the egg without any sensory interaction. They can detect light, or a horse can run after the mom within a few hours, or even a whale follow the mom. But it takes us huge amount of time to learn how to eat, walk around. So our development is very, very protracted. And this is the price we pay to develop a superior nervous system, because you need to have this kind of environmental interaction with your hardware, which was produced in utero, to have a hardware which was partially developed through the signals which you receive from the environment. So if we look at our closest relatives, you can see that their brain development is pretty much done by 2 years of age, whereas, as I mentioned, our peak is around 11, 12 years of age, and after you have a decline in connectivity and cell numbers. So this is why we have so many problems with brain development. Because if you have a slightly altered hardware, your cells are produced in different proportions, or they cannot interact with the environment in the proper way, then you can develop like schizophrenia or autism or learning difficulties, which will have an impact on your life and also your surrounding, your family, and also society. So these conditions, sometimes they produce very little anatomical abnormality, and they have a complex origin. And sometimes they manifest decades after the insult. So for instance, if you can have very early environmental interactions and combine this with genetic factors, can have an effect on the fetal growth and also brain development. So I'm talking about also these interactions with infection, diet, or even the gut microbiota of the mom. And these, in combination with genetic susceptibility, could actually lead to other subtle alterations and conditions like attention deficit hyperactivity disorder, or autism spectrum, or even childhood epilepsy. Childhood epilepsy is just as common in kids than later on in the adults. So it's about 1 in 200 in the population. And some of these early developmental steps are also beginning to be linked to anxiety, depression, schizophrenia, and I would go even further, that it can also lead to obesity, diabetes, hypertension, metabolic disorder, cardiovascular diseases. So right now, most of the funding is going to Alzheimer's disease, Parkinson's, but I think within the next few decades, I think these early steps will be in the focus, because some of these conditions can be explained with early interactions. So even Alzheimer's disease, Huntington's disease, now they are linked to very early steps in development. And also, if you look at the ranking of the costs of diseases, and you use this scheme, the disability adjusted life year ranks, you notice that the economic damage condition is causing is very high for developmental conditions. So for instance, preterm birth complications are very high, was very high on the list, and also neonatal encephalopathy, and it didn't change too much within 20 years, and even now they are very high. So that's why I think we need investment in brain developmental abnormalities and conditions. So if you have altered hardware to start your life, then you will have a bit more difficult time. I'm not saying it's impossible to recover, but everybody has the right to get a functional nervous system to start their life. So let's go through some of these conditions, what can go wrong. And I start with neurogenesis. So most of the cells are born in the lumen of the neural tube. You can see that this is the inside of the tube, and this is where you have the cerebral spinal fluid filled cavity. And you see that most of the divisions, dividing cells on this time lapse, they divide next to the lumen, and then they migrate up below the PL surface, below the skull, to form this first postmitotic cell layer, the so-called preplate. And every single dot here is representing the dividing cells. So you can see that most of the divisions are in the ventricular zone, but you also have some divisions in the so-called subventricular zone. And initially, we thought about 25 years ago that these processes were from radial glial cells, so supporting cells. And it turns out, around 2000, it was discovered that these are actually radial progenitors, which can produce directly some neurons, or they can produce some other intermediate progenitors, and they are basically progenitor cells for neurons. So the name is wrong. We still call it radial glia, but they are, we now know that they are not glial cells. And these radially oriented neural progenitors, they can produce neurons directly or indirectly. And these intermediate progenitors, they are very prominent in primates, especially in human, because we have a bigger brain, and we have to produce this brain much faster. So obviously, this is a very vulnerable cell population, and I will show you that the Zika microcephaly, the virus was acting on these cells when they caused microcephaly. So right now, we believe that there are three progenitors which produce neurons. So these radially oriented progenitors, these intermediate progenitors in the subventricular zone, and then the outer subventricular zone intermediate progenitors. And they all have slightly different movements because the centrosome is, which is necessary for the division, is located in different locations. So these radial progenitors, the nucleus has to drop down to the end feet, and then they can divide. The intermediate progenitors in the subventricular zone, they don't have to move anywhere because the centrosomes are in the cell body. And then the outer subventricular zone cells, they have to be elevated. So they have a different interkinetic nuclear migration than these other two. So this is a highly complex interaction, how our neurons are produced, and almost all of these steps can go wrong. Let me just show you two. There is a gene called TBR2, which is expressed in these intermediate progenitor cells. If there's a mutation in this cell group, then you end up with a brain very similar to the one I showed you from Willis, polymicrogyria syndrome, and you have more but smaller South-Scion gyri, you don't have corpus callosum, and also you have learning difficulties and also intractable epilepsy. So this gene, which is involved in amplifying the output from the progenitors, is vulnerable for this mutation. Or the Zika virus, you remember a decade ago when it was sweeping across Latin America, it caused this devastating effect in the first trimester because Zika virus attacked these intermediate progenitors. And in some cases, it manifested as microcephaly. So many of these neurons, when they migrate, they are born away from the location where they have to migrate, so they will have to get into position. And there are lots of problems with this neuronal migration because you need a cellular molecular interaction with lots of molecules to get to the position. And this can be disrupted. So here you can see that some of the cells, they migrate from the ventricular zone to the surface. So these progenitors produce the cells, and they have to drag themselves up to the PL surface. And if they get stranded, then they will not end up in the right location. So these are the glutamatergic excitatory neurons. The GABAergic neurons, they have a different origin. They have a tangential migration. So they move this way, whereas the radial migration was up from the ventricular zone to the PL surface. So both types of migration can go wrong because of genetic or epigenetic reasons. So I just show you a couple of examples what can go wrong. So you have these over 200 different cell types in the 6 layers of the mammalian cerebral cortex, and it does make a big difference whether the cell can migrate to the destination or not. So when the cells are born, they already have an address, they have a destination. They are committed to that fate. If they cannot migrate, then it will cause big problems with the established circuit. So what can go wrong with the migration? So it was shortly after Hiroshima, where the atomic bomb was dropped first, that they noticed that some of the women in particular stages of pregnancy had kids with brain developmental disorders. So let me just show you a brief video which is explaining this situation. So we might need sound for this.
The bombs over Hiroshima and Nagasaki killed up to 250,000 people. Amongst the survivors were more than 1,000 pregnant women whose unborn babies were exposed to radiation. Clues began to emerge that some of the babies had been passing through a critical stage at the time of the blast. Radiation interrupted the conversation between glial cells and migrating neurons. This is Narumi Toda. She was born on February 2nd, 1946. 7 months earlier, her mother had been half a mile from the Hiroshima bomb. Narumi is retarded. William Schull has been studying people like her to learn about radiation's effect on the brain.
Well, the story about radiation-related brain damage actually began here shortly after the bombings. We soon recognized that children who had been exposed in utero, a small number of them, were severely retarded. The earlier analyses of the data tended to focus on a conventional division of the gestational period into trimesters. We divided the gestational period into 4 intervals of time, which correspond to more or less separable periods of development of the brain. Once this was done, it became very clear that 80%, in large measure all of the cases of mental retardation, occurred to women who were in a specific stage of their pregnancy. That is, the fetus was between 8 and 16 weeks following conception. Now, we know from neurobiological evidence that that period is extremely critical because 2 fundamental events are occurring.
First, neurons are being created at the astonishing rate of 250,000 per minute. Second, the glial cells have formed a precise pattern of fibers. The neurons crawl along this scaffolding to their final destinations. The bomb's deadly radiation interrupted millions on their long journey.
Neurons have a specific date of birth and they'll have a specific destination. And they do not migrate haphazardly. They have an address and they move to that address in a highly choreographed sequence of events. And since we know that brain function critically follows brain position, cells have to be in the right place to do the right thing. Even though alive, even though in the general area, they don't perform the proper function if they're not in the proper site.
So that is the very big problem with these brain developmental abnormalities, that if the cell cannot make it to the destination, then you will have an altered pattern and an altered circuit, which might not perform the same way. Of course, the brain can compensate for a large extent to all these, but if you can't make it, then you will have some cognitive issues. Now, to extend a leading edge of a migrating cell, you need certain molecules. actin, filamin, and many others. And then you have to drag the nucleus. And for that, you need different molecules as well. So you have this perinuclear cage, which you have to drag. Now, you are not going to be surprised to hear that any mutation in this network or here will lead to mental retardation or learning difficulties or epilepsy. So basically, you have to deliver the cells to the right position. And now we're beginning to understand the molecular nature of these. So let me just show you some of the very common developmental abnormalities which can occur. So most of the cells, as I mentioned, this is the normal situation, are migrating out, and they form first the subplate layer and the marginal zone, and then they get split into two, and then you have an inside first, outside last development of the inner and outer layers. So you will have layer 6, 5, 4, etc., like this. Now, if you have problems with the leading edge extension, so some of these molecules are missing from this part, then you get a couple of cells piling up in a random fashion next to the ventricular system. So let me show you a couple of MRI images of these. So here you can see that these cells couldn't move from the ventricular zone, and you will have islands of gray matter, and they can produce epilepsy and also learning difficulties. So these are called the periventricular heterotopias. Or you can have nodular heterotopias. So you have a couple of nodules here which couldn't make it to the cortex. Sometimes the lower layers make it, but then the upper layers pile up below. And that is causing a smooth surface brain, and that's called type 1 lysencephaly. And the smoothness of the brain can vary. So this is not so severe, but this is a quite severe lysencephaly. Lysencephaly is just an expression saying that smooth brain. But basically, you can have many different grades, and depending on how severe the abnormality is, you call it differently. You can also have completely reversed cerebral cortex. So instead of inside first, outside last, you have an outside first, inside last reversed, and that's called the Reeler phenotype. The name came from a rat mutant, which was showing ataxia, reeling, because of cerebellar abnormality. And in fact, in human, they also identified such mutation in a Saudi family, where you can see this lysencephaly and also the reversed cortical organization. Sometimes cells, they don't stop in the right place. They go through the external limiting membrane, and they have islands of heterotopic cells in the surface of the brain, and that's called type 2 lysencephaly or cobalt stone lysencephaly. So I think you get the idea that how these migration disorders can cause a cortical dysfunction, which manifest in cognitive disorders. So now we begin to understand the genes and also where these genes act. Now, for some of you who are engineers and dealing with physics, I would like to demystify this whole brain folding business, that you don't need a living structure to model this brain folding. Just the expansion of the material in the brain is sufficient. So we call these brain developmental abnormalities based on the shape of the brain, and nothing changed since the time of Willis. Neuropathologists, they still call it like this. So basically, they just give Greek and Latin names to the shape of the brain. But now we know the molecular background. And Tallinen, who worked at Harvard, but having Finnish origin, he modeled this by looking at the rail brain, human brain, and made a plastic model of a 22 gestation week-old brain. And this plastic model was expanding when you put it into fluid. And in addition to this, Tallinen also dipped it into another gel, which was expanding a little bit faster than the core gel. So we have only two layers in this, one inner and one outer. And then he placed it in a solution, and you can see that you could recapitulate the formation of the sulci and gyri in the dish. And I'll show you a video which is accelerated to how this model was expanding. And you can see that this is the lateral sulcus, this is the central sulcus. You're beginning to see exactly the same sulci and gyri you would see in a real brain. So you don't need living structure for this. You can model this just with the expansion. And it's frighteningly similar. If you start with this model, frighteningly similar what you can actually see with the imaging brain. So if we want to understand how these brain developmental abnormalities change the shape of the brain, then we actually have to understand what is the rate of neurogenesis and migration and what are the forces which shape the folding of the brain. So, and this is how we can understand what's going wrong in polymicrogyria syndrome or lysencephaly. So if you look at the branches of evolution and you compare different species, you can find primates with smooth brains and also rodents with sulci and gyri. So this is something the nature is using in all branches of the animal kingdom. So it is not primate specific to have a brain. However, it is spectacular that if you look at the primate brain, you have so many progenitors. Every single dot here is an intermediate progenitor in the inner and outer subventricular zone. If you look at a rat or a chicken brain, you see very few cells. Or in a turtle, you only have just inner ventricular zone rich progenitor population. So probably the brain folding is something to do with the velocity of the development of the brain. And in primate, and especially in human, you have lots of progenitors and they expand faster. However, it is not true that this is primate or human specific. My group looked at lots of South American rodents, agouti, capybara. They have folded brains and they have roughly the same proportions of the progenitors. So my friends, Wieland Hüttner and Arnold Kriegstein got it wrong in these papers. So it's not a primate-specific feature to have these progenitors. So the maternal environment can also have a huge effect on the brain development, and there is a very intimate interaction between the baby and the mom. You have the placental barrier in between them, so only certain agents get through this barrier. But if the mom has an inflammation, the cytokines can very easily go to the baby, and the cytokine receptors are expressed in the progenitors. So this blood flow, nutrients, hormones, cytokines, serotonin, they can all have an effect on brain development. And also some of the damage is self-imposed. So alcohol consumption, cannabis or cocaine can lead to brain developmental abnormalities. And one of the most common ones is actually the fetal alcohol syndrome. So this syndrome was discovered in Seattle by this group. So I let them to explain what exactly how they discovered it. Again, I think we will need some sound for this.
A portrait of despair. Among its horrors, a mother pouring gin into her baby. But now we know that even the drinking of a pregnant woman can affect her unborn child.
Fetal alcohol syndrome was discovered by a resident in our department of pediatrics in 1970. Christy Hewland was doing a study of infants who were small for gestational age, infants who were born on term but were little. And among those children, she found a few children who looked alike, who were very small, who were all retarded, and whose mothers all had a history of heavy alcohol use.
The report met with skepticism. If alcohol was the culprit, when and how did it do such harm? Baby monkeys helped Claren's team find the answers. This monkey is developed normally. He can remember there's an object in the box, even when he can't see it. Could monkeys who were exposed to alcohol when they were young embryos do the task equally well? This one shows no interest, a sign that the frontal lobes of its brain are damaged. But when does the alcohol have this deadly effect?
There's a good deal of evidence that exposure to alcohol is bad for the developing brain throughout gestation. But what this particular study suggests is that early exposure is in fact very important and could cause significantly more problems than higher exposures later in pregnancy do alone.
In Gothenburg, Sweden.
So that's the problem with the fetal alcohol syndrome, that once you finish producing the brain, then there is no way back. If this is the hardware you could produce, then it's very difficult to compensate for this lack of cell numbers, connectivity, cell types. You can improve the condition about when you get it about the age of 10, 12, but you will have a very altered, disrupted life and upbringing. And the severity of fetal alcohol syndrome is usually measured from the facial abnormalities. So this is how they measure it, and they have different categories, you know, the philtrum, the upper lip, and all sorts of, so they use AI now for all these. These are the measurements they make. And the problem with fetal alcohol syndrome is that it is, in some parts of the world, is actually extremely common. I have a friend in South Africa, Professor Newton, who said that it's 1 in 10 in certain parts of South Africa, the fetal alcohol syndrome, because they pay people with alcohol in some plantations, and that has a devastating effect. But even in the Western world, you have 1 in 100, which is actually, of course, it's a spectrum, but it was surprising for me. So I kept reading about this, and what is even more surprising is that if you go into prisons and you look at some people who couldn't fit into society, they ended up in prison, you can have quite a high number of fetal alcohol syndrome victims in a way. And a large proportion of them will not be able to conduct an independent life. And I don't know what is the cost of incarceration here in Lithuania, but in the States, to keep somebody in prison or in a young offender place is $120,000 a year. So you could spend that to educate somebody at Harvard for two of them, actually on the same cost. And after fetal alcohol syndrome, you don't get better. So this is a condition which is will not go away. And if you look at the incarceration rates in different countries, there are some surprises, especially here if you look at Louisiana. So this is a problem which you don't hear too much about, but I think this is an existing issue to discuss. And now we are moving into my research area, which is the cortical-thalamocortical network. So I showed you this diagram before. So you have lots of projections going into the cerebral cortex from the thalamus. Almost every sensory information you receive from the environment, with the exception of olfaction, is going through your thalamus. And then it's relayed to the cerebral cortex. And in return, so all this information is going through the thalamus to your cerebral cortex. But in return, layer 5, 6, and 6B sending back projections to the thalamus, and it's regulating what input can reach your cerebral cortex. And just to illustrate the scale of things, these projections back from the cortex, they outnumber the afferents to the thalamus 1 in 10. So most of the time, you are listening to your brain and not your environment. You have your own image of the environment, and occasionally you update it with your sensory input. So that's why these projections are very important in regulating arousal, attention, sleep. And And as I mentioned, half of our brain is right now in sleep, but which part is determined by these projections from layer 5, 6A, and 6B. And these cells in 6B are particularly interesting because they are largely transient. Only about 20% of these cells will survive, but they have a very powerful grip on the regulation of these connections. So to explain what I'm working on, I have to introduce the term of the first order and higher order thalamic nuclei. So most of the sensory input, like vision, is mediated through first order thalamic nuclei, and then they go to first order cortical regions. And there is, in the primate, there is a huge part of the thalamus which is not receiving direct sensory input. They receive the input from the cortex, and they relate this information back to other cortical areas. So you have a transthalamic cortico-cortical communication. And you only awake this system when you want to pay attention to something, or something more challenging, or you want to learn a new task. And you use this higher order thalamic system sparingly. But during evolution, this is the part of the brain which expanded the most. Like, think about human pulvinar. This is much bigger than in any other species. So I'm interested in the interaction between these layer 6B cells and waking up this higher order system. And I believe that this 6B system, if you have altered survival or distribution of these cells due to brain developmental abnormalities, then it will regulate an abnormal arousal, sleep system and perception, and that's what we see in schizophrenia and autism. So I just show you the evidence why this layer 6B cell group is so important. So the reason why I studied this cell group, because I was very interested in the developing brain, and I could follow their role during early stages. And these layer 6B cells are very, very prominent during early development, but then they disappear. So they were considered transient cortical circuits. And the remnants of these cells are in 6B. So you remember I showed you that most of the neurons which are born initially form this primordial plexiform zone with the first postmitotic cells, and then they split into marginal zone and subplate by the later arriving cortical plate cells. And then you have the cortical plate developing in an inside first, outside last fashion. But then these cells, these subplate cells, they orchestrate the cortical development. They have multiple origins. Some of them migrate in tangentially, others radially, so lots of things can go wrong. And they are the first cells which receive the thalamic input, and they also guide the thalamic projections to the cortex. Without this interaction, the thalamic projections cannot get to the cortex, so they have a very important guidance role early on. If you mutate some of the genes in these early corticofugal projections from subplate, then the thalamic fibers are abnormal. In human, the subplate cells are huge. They express all sorts of proteoglycans, and they have very high water content, so you can image them with MRI very easily. So the first thalamic projections, when they reach, so this is a human brain where Sarah labeled the fibers with carbocyanin dyes, so they passively diffuse in the lipid membrane, so you can use postmortem brains to study these projections, and you can see that these projections invade the subplate and also some of the germinal zone, and they have an input of the area differentiation. And these are the first projections which also deliver the activity patterns to the cortex, and you can see that the first depolarization pattern will occur in the subplate when you stimulate the thalamus, which will carry the sensory input to the cortex. And later on, this activation pattern moves into the cortex, and the subplate will be kicked out from the circuit, and now the thalamic fibers target the ultimate parts of the cortex. So you have transient circuits the thalamus is first targeting the subplate, then the subplate is feeding into the cortical plate, and then you have a transient circuit with the ultimate target cells, and then subplate will die off, and then you have an adult circuit. So this took us 20, 30 years to understand this circuit, but now we know. And we also know that if you alter these circuits, then you have more subplate cells surviving. So I would like you to think about the subplate as a transient dynamic scaffold. I don't know whether you have been to St. Louis. They have an arch. I've never made it up there, but I've seen the arch. And guess how it was built? It's not an easy task, an engineering task, to build this arch. So what they did is they started on the two sides, and then they introduced a transient scaffold between the two, and then they kept going up until, and they moved the scaffold up until they lifted the last piece to the top. So this is the moment when they were lifting this very top here. But after this, they removed this transient scaffold, which was integrating into the building during the development. So I want you to think about subplate as a dynamic transient scaffold, which is essential for building the brain. And if you don't have these cells, then you cannot finish the building work, just like you couldn't finish the arch. So if you remove subplate cells, you cannot form barrel cortex or ocular dominance columns. You don't touch the cortical plate, you just touch the subplate. Nevertheless, the thalamic fibers cannot sort themselves out. So that's why it's very important that these cells stay alive. So we have known this for a long time, that many of these subplate cells, they are very numerous at birth. This is a cat cerebral cortex. If you birth date them with radioactive thymidine, you can see that you have a lot of cells still in a newborn, which were born very early. But if you wait a bit longer, then you see that many of these cells are gone, and you only have just a few interstitial white matter cells remaining. So they are transient. But if you do a similar birth dating study for layer 6, which is adjacent to these 6B cells, then you see that they survive in roughly the same numbers. So these cells are special. They are only present for certain parts of development, and then they disappear. And the same is true in macaque and also in mouse. So we study these cells, and we want to understand what is the function of the remnant cells, and why are they changing in certain cognitive disorders. So in schizophrenia, you have more subplate cells surviving in the prefrontal cortex. We don't know why, but they might be responsible for some of the cognitive disorders, and that's why I gave the title Shadows of the Subplate, because in autism and schizophrenia, you have more of these subplate cell remnants lingering around, and they can have an effect on the cognitive dysfunctions. So my group is looking at what is the reason why these cells cause cognitive disorders. So the theory is very simple. You have more subplate cells surviving if you have altered development due to in utero infection, inflammation, drug exposure, or hypoxic ischemia. And then these altered cells will integrate into the cortico-thalamocortical loops, and that will have a different effect when they normally disappear. So what is my evidence that they actually cause some cognitive dysfunctions? And basically, we use very specific mouse lines where we can modulate and monitor the function of these cells in a very, very specific manner. And I will show you some results showing what is the connectivity of these cells and also what is the function when they get altered. So this line has the layer 6B cells and their projections labeled to the thalamus. And you can see that they selectively target the higher order thalamic nuclei. They don't go to the first order thalamic nuclei like LGN or VB. and they are very different from the adjacent layer 6 projections. They have very widespread connectivity in the brain, so we trace these up, and also they have very specific relationship to some of the inhibitory neurons in the thalamic reticular nucleus, and this is very important for sleep regulation. That's why I show it. They are very small, they have very small boutons compared to layer 5, and they basically involved in the regulation of these higher order thalamocortical interactions. So we trace the input to these cells, and we know who is talking to them. We used rabies virus to trace the projections. So rabies is jumping synapse, and we used a strain which is only jumping 1. Otherwise, you would wipe out Oxford, basically. So this was actually San Diego. This was established by Ed Callaway at the Salk Institute, this method, and we just use this. So we discovered that some of the hypothalamic cells have a direct input to these layer 6B cells. And interestingly, the cell group which was talking to these cells was in orexin positive cells. So orexin is a peptide which is produced by the lateral hypothalamus. And if you don't have this peptide, you have cataplexy. You probably have seen videos of dogs when they get excited and then they fall asleep in the middle. So basically, we discovered that these layer 6B cells get orexin input directly from the lateral hypothalamus. And what is also interesting is that if you record from different cortical layers, they are not responsive to orexin, with the exception of layer 6B. So this system is very important in regulating arousal, attention, and also sleep. So we wanted to know how would the phenotype change in these animals if we silenced layer 6B, which is the target of the orexin input. So what we did was we genetically engineered a mouse line where we silenced the layer 6B cells, these ones which have the cell body here up in the cortex and their projections in the thalamus. And we wanted to see how this will change the behavior of the animals. And to reduce the activity, we basically use the same techniques as you use when you inject Botox. So basically, we just inhibited the release properties of these cells so they could not talk to the other cells. That's what you do when you apply Botox to smoothen your wrinkles. So basically, we removed SNAP25, which is a protein essential for the release of the synaptic vesicles. So all the cells where you don't have SNAP25, they cannot talk to the other cells. And then we looked at the effect, how this will affect this network of connections. And we were surprised to see that the animals were all alert, no problems with movement, no cataplexy. They had normal locomotor activity. They had normal circadian rhythm. If you put them in constant dark, they behave the same way as the controls. And if you gave them a 6 hours jet lag, they also responded the same way. But what was very interesting is that we almost completely removed some of the sleep spindles from these animals. So they had a very specific sleep abnormality, which is very different if you silence other cell types in the cerebral cortex. And what was also very interesting is that these animals had no fear. We reduced the anxiety of these animals. So they are going out to the open arms of these elevated plasmase, which normally mice they don't like. And they also go to the light side of the light-dark box. And also they had reduced novelty preference. So normally when you put an animal into a Y-shaped maze and you block one arm, after a while they habituate to this. And when you reopen the arm, 80% of the animals will go to the novel, but not the DRD1A, Cree, SNAP25, silence layer 6B animals. They still went 50-50. So we are very interested in the possibility that these layer 6B cells, especially in the prefrontal cortex, they can be targets of the lateral hypothalamic orexin, and perhaps this can mediate anxiety it would be really good to target this, because then you could actually interfere with anxiety without changing all these other functions orexin might have. So we are going after this cell group, and these cell groups in the prefrontal cortex are responsive for orexin, so the mouse does have a, Fernando and Rajiv have been looking at the sensitivity of these cells using multi-electrode systems. So you have thousands of electrodes, you have a brain slice sitting there, you pour orexin on them, and you have a look which site is active. And indeed, prefrontal cortex has the greatest sensitivity for orexin, but if you remove SNAP25, then this response is completely abolished. So we are now looking at how we can modify this circuit in a more selective manner in space and time, and how this will affect the behavior of the animals. We also looked at human brain tissue, and we confirmed that the equivalent cells have orexin sensitivity in human epileptic tissue. The white matter interstitial cells have that. So we are interested in the possibility that these cells, the shadows of the subplate, have a long-term influence on the arousal, anxiety, and some of the symptoms which you see in autism and also schizophrenia, where you have altered numbers of these cells. And we believe that studying these cells could link some of the brain developmental abnormalities to the symptoms you see in later stages. And this is all related to these thalamocortical and corticothalamic systems, and I think this is changing the balance between them. So if you have more cells surviving, then your whole cortico-thalamocortical communication can change. Your sensitivity, all these vital neuromodulators can change, like orexin, dopamine, neurotensin, and we are studying this. We also, I have affiliation to Berlin, to the Charité. They have these Einstein visiting professorships, and what we do there is we keep some of these subplate cells surviving longer by knocking out members of the mTOR pathway, so P10 conditional knockout, and basically we are looking at the behavior of these animals when you have more subplate cells surviving. So unfortunately, this is not our department, this is biochemistry. Physiology is in this old department, and my lab is here in the ground floor. And I'm very grateful to all the collaborators and also lab members and also the funders, and also to you for your attention. Thank you very much.
Thank you for a very nice talk. And now we have a few minutes for questions. So maybe we have questions from audience.
Okay, thank you. Very nice talk. I have a small question. There are some, I know some friends that had epilepsy, but they developed it during the puberty. So I'm interested what's happening in there, since epilepsy can start, you know, when you are born and you are very unlucky in that kind of regard. But I'm just interested what happens in the brain during the puberty, and why brain starts to do zaps, and yeah, it would be interesting.
So epilepsy is a huge topic, and there are lots of different forms of epilepsy. And so I don't know specifically this particular, but there are two major groups. And one epilepsy, which I believe is related to these functions, is the absence epilepsy. So that's a particular type of epilepsy where you don't see much on the patient, so they don't have these clonic movements, but they just lose a bit of attention time to time. So you have a kid who is just not paying attention for a few seconds, and then they get back. So that is also epilepsy, and I believe that this system I was talking about is probably related to that, but there are many other types. But your question was, what's going on during puberty? So huge amount of pruning is going on. So after 11, 12 years of age, you have a huge decline in synapse numbers. So that has a huge impact on the circuit and the balance, and maybe that can trigger the manifestation of these abnormal activities. And this is also the reason why some of these cognitive conditions also manifest later in life. So the schizophrenia, you don't see anything until, maybe the end of the second teens. Nothing before. Although some people say that you can maybe see some arm swing changes at younger stages. But I think these are anecdotal at the moment, so we don't have long-term effects. So I think puberty is the time when you have a huge amount of remodeling of your brain, and maybe that's why some conditions manifest. Yeah, manifest then.
I was wondering about the connection to autism, because as far as I know, there's like a huge amount of autistic people that are very good at certain things, maybe a small amount of things, but how is that connected? Because apparently something went wrong, but how do they turn out to be so incredibly good in certain aspects?
So, it's also a very, very complex topic. So what I wanted to talk about is that in autism, you can have sometimes very focused and very specific issues with attention. Sometimes they really pay attention for certain things which you wouldn't, and they don't let it go. And I think the circuits I'm talking about could explain some of it, why you don't suppress some of these higher order circuits and say, fine, I know roughly what's going on, so I don't need to pay more attention to the details. And maybe if you have an altered layer 6B circuit, then you keep thinking about these and keep waking up the higher order thalamic systems. And also, it could relate to anxiety, because if you think about it, sometimes you keep thinking about minor things happen to you, and then they cause anxiety to you. But most of the time, you just say, okay, fine, I know what was going on, so I suppress. So the balance between this first order, higher order system could explain maybe these symptoms. So that's what, but it's, imagine to study this, very difficult. So now I have to move to behavior, where I'm not feeling at home. So first of all, I had to move to sleep. But that was okay because we have a very, very good sleep researcher, Vladislav Vyazovsky, in Oxford. He did his PhD in Zurich, then worked with Tononi in the States, and now he was recruited to Oxford. So he's a fantastic sleep researcher. But now for the behavior, it's very difficult because I would even like to study not just attention, but illusion and delusion. Because if my prediction is, if you wake up this higher order system, you will suddenly see things which you shouldn't. So we are planning experiments on this by inactivating this layer 6B system.
Thank you.
Okay, more questions? Yes.
You said that at the age of 11, this restructuring happens. What is the reason for it?
This pruning we were talking about. So
yeah, I understand, but why does it happen evolutionary that at the age of 11 we start having this pruning? What happens at all?
I exaggerated a bit. So basically, it's not happening exactly at 11. If you do a chronic imaging study on the brain and you color code the areas which expand and which regress, it's actually a wave. Actually, starting from the back, you start losing cells there, and then by the end of the puberty, you begin to lose also frontal lobe. But frontal lobe is the area which is growing the longest, and then it's also, and from the pattern you can see from this chronic imaging, you can actually predict some of the cognitive dysfunctions. So if you image a child, it's very rare to do MRI for every year for a child, but if you do measurements, you can actually predict who will have schizophrenia and who won't. So Toga did it in UCLA, these studies. So basically this 11 years of age, you have to take it, so it's a wave of activity, and different cortical areas have a different peak. So it starts in the back of your brain, and then it's getting thinner in the front.
And Mars, why it starts at all? Like, do the cells just start dying because they're too old? Like, why there is this process happening at all?
Why not keep it?
Yeah. Why doesn't just grow and then stay and is happy? Like, why there is this wave happening at all?
It's a very good question. So maybe you don't want to have all these connections once you established and you have a functional nervous system. So initially you need to try out lots of different permutations, and you shape your circuits before puberty and during puberty. And after that, maybe it's advantageous to freeze that situation, and you have a very functional, efficient nervous system. But it's also very bad. Many of you probably do skiing or tennis. It's hopeless to do it after puberty if you want to be a pro. So by the age of 12, most of the repertoire of tennis player is established. If you didn't get it, that's it. You are not going to become very good. So I started skiing very late because Hungary is very flat. So and I will never become a good skier. So my kids from a Swiss mom, they never want to ski with me because I'm very slow. So from the age of 5, they were skiing. So when they prune the connections, after that you established all these movements and autonomous motor programs. And after that, you just work with the bare minimum. So you cannot really adapt to why evolution is doing this, I have no idea. Yeah. But maybe you could produce some transgenic animals who wouldn't have this and see whether this is advantageous or not. Yeah.
Thank you. Fascinating, really. I think I even lack the vocabulary to ask the questions. But one thing that stayed in my mind was about the whole sort of importance of migration, among other things, of course. I'm just trying to understand, what is the biophysics? So you mentioned it, and also in the film, they talk about it getting an address and a source and a destination, etc. But what's the biophysics that this migration takes place so, in the, let's say, normal brain, that has happened so, I would say perfectly. And that's the radial kind of migration you showed about, but there's another one which looks to be even more complex, right? You have to follow a circumferential kind
the tangential
of
for the GABAergic cell.
How does it work?
So it took a while to understand that when you have a progenitor cell and it's entering the last S phase before it's producing the cell, the combinatorial transcription factor cocktail, which is expressed in the cell at that time, will determine the fate of the cell. So the gene expression in the ventricular, subventricular zone is changing all the time. And this is how you generate cell diversity and variety, by segregating cell division in space and time. So once these cells leave the S phase, they have an address, they have a fate, and they have to make it. And they rely on these molecular mechanisms, which I outlined, the leading edge extension, the nucleokinases, and then also they have to stop. And if you don't have that readout, then you will have a problem. So that's why it's so vulnerable, this whole system.
But you didn't speak about
the tangential. That's also a huge topic. So basically you have 3 ganglionic eminences, the medial, lateral, and caudal ganglionic eminences. They produce different GABAergic cells. There are about 20 different cell types. And it's interesting that if you alter the proportions of one of these GABAergic interneurons with a little percentage, you end up with epilepsy. So it's almost better not to have a cerebral cortex than a cerebral cortex which has a minor adjustment of these interneurons. So if you produce a mutant mice where you alter these GABAergic cells, which go tangentially a little bit, like NPY, somatostatin, parvalbumin, then you can have epilepsy by the age of 3rd week of that mouse's life. So with very severe symptoms. So it's an amazing system that you have to produce this hardware in the right proportions, and then you have to have the interaction with the environment to function. So it's, in a way, it's a miracle that we don't have more developmental abnormalities. Yeah.
Thank you.
Okay, I would like to thank again Zoltán Molnár for a nice talk and discussion. So please greet his with applause.
Thank you very much.